Okay, so yeah, I'm thrilled to be here to present on some of the stuff that you guys have been learning about and how it's being applied in a very real way to help protect Canadians through our national SARS-CoV-2 genomic surveillance program. I have a lot of slides, so I'm gonna just dive right in if I can. Okay, so I thought I'd just start it off by showing you how we actually do SARS-CoV-2 genomic surveillance. So it all starts at the testing, right? So if a person is feeling sick, they go into a clinic or a hospital, they get a swab. That swab is stored and then transported to a laboratory that has sequencing capability, the pathogen and its genomic material is extracted. In the case of SARS-CoV-2, which is it has an RNA genome, we're extracting the RNA. Then so there's a step where we convert it to DNA. And then we run it through a sequencer that generates raw genome sequence data, and then we use bioinformatics to assemble that into a pathogen genome, and then we perform an analysis of the genomic sequence data. One of the things that I wanted to point out, I hope to touch on it later, is that there's a special protocol for sequencing the SARS-CoV-2 genome. It's called amplicon tiling. The problem here is that the specimen as received it contains a combination of human genomic material and the viral genomic material, and it can't really isolate that viral genomic material out before sequencing, and so the amount of viral genomic material is very small relative to the amount of human genomic material. So what we do is use this tiled amplicon approach where PCR primers are designed to target the genome, SARS-CoV-2 genome specifically, and then amplify a region that can be sent through the sequencer. It's about the right size to be able to be sequenced by a sequencer like an Illumina sequencer or an Oxford Nanopore sequencer. So about 400. So the Arctic protocol is a common, popular protocol, and so uses these. It generates 400 base pair overlapping amplicons, and then we use those overlapping amplicons to reconstruct the original genome. Yeah. So why do we do it? There's a number of really important reasons for doing SARS-CoV-2 sequencing, and clearly, it is a fundamental pillar of the national and global response to the COVID-19 pandemic. And here's why. So it allows us to track the transmission at regional, provincial, national, and international scales. And I'll show you how that's done. It can assist in outbreak investigations by identifying the origin and spread within a given outbreak. For example, if you're trying to track a super spreading event. You can monitor the effectiveness of our mitigation strategies, such as border closures, by identifying new introductions of the virus into Canada, and how is that how does our policies influence the ability to mitigate the introduction and spread of the virus in Canada. It also allows us to perform situational awareness of the circulating variants. Essentially, we want to know which ones are in the country and how much of them are there of each subtype, and are they growing or shrinking? And it also allows us to detect viral characteristics. These are important. These are actually what determine things like a variant of interest or variant of concern. They allow us to detect viral characteristics that can impact our detection methods. So by identifying mutations that might reduce the effectiveness of our standard PCR-based tests, clinical outcomes by identifying the variants that may be associated with a severe disease, and natural and vaccine-based immunity. So, but more importantly, the vaccine effectiveness by looking to see which sequences are susceptible to the immunity that is induced by vaccines, or maybe ones that might escape the immunity that's induced by our vaccines. So when we were running at full stream, which was for about a year and a half, we had 17 different labs performing the sequencing. Right now it's fallen to about 10 core labs, but at one time we had about 17 labs around the country. And so that includes public health labs like the lab that I work at, which is the National Microbiology Laboratory. But other provincial labs, our counterparts in the provinces, we call those, we'll call those the Canadian Public Health Laboratory Network, and that does the bulk of it. But also we have other large-scale sequencing partners and some hospital diagnostic labs that are participating in the sequencing. So it's a really large-scale activity. And lots of money and lots of effort has been invested into this program. So here's a little bit of the performance of our sequencing program. So when we first started, which was in April of 2020, just after the onset of the pandemic, there's a lot of work required to just deploy all of the equipment and the training and funding for the reagents and developing standard operating protocols and a lot of that early work. And so the numbers of sequences that we were generating for the first, well, throughout about 2020 was, well, enough to be able to do genomic surveillance, but it was relatively modest. But through our capacity building effort, we've managed to really increase our throughput substantially. So whereas we were averaging under 5,000 sequences per month for 2020, after that we ramped up to where we can routinely generate about 25,000 sequences per month. And if we need to surge, for example, if there is an emerging variant, Omicron being a good example, we could surge up to about 35 to 40,000 sequences per month. And that's what's shown at this little histogram on the left. On the right, these are the collection to sequence times. So sequencing is not easy. In fact, it's very difficult. And it involves a process of getting the test, getting it transported to the lab, getting it prepared and sequenced, and then it has to be transferred up to the national database at the NML, and then also up to the public archives. And so at the top, you can see our median collection to submission times. And they were quite long for the first, they were lengthy, up to about somewhere on the order of about 90 days, so about 3 months to get on average, to get all of the sequence data transferred up into public archives, like the GISAID public archive. And so it's on a log scale at the top there, just so that you can see it, but we have improved substantially. So if you look at the bottom, on the bottom right, for example, over the period of mid-April to mid-May, our collection to submission times have dropped to under 3 weeks and are close to about, and currently are close to about 2 weeks, which makes it useful for performing genomic surveillance. So it's an effective and fully operational genomic surveillance program. Okay. Along with the sequence data, there is metadata. And so as the VADA group, I'm sure is all aware of what metadata is about, the data that is used to describe the sequence data. And so this includes things like technical data, like what sequencing protocols did you use, and also can include patient data. And because we are collecting from so many different sources, it is important to establish a standard, a metadata standard. And that was one of the first things that we did, is to establish the metadata standard so that when the data is collected, it is harmonized and usefully described. But in order to ensure that that is the case, we have to do data validation, and to do that, we developed a tool. Actually, our collaborators from Simon Fraser University, Will Shaw and Emma Griffiths, developed what's called a data harmonizer tool, where you can load the data, and it will go through and identify errors in the data, and essentially help you to clean it up and to validate it before it goes into the national database. So once it's in the database, then we do we do our analysis, and then we also we submit it to these national repositories like the GSAID National Repository, NCBI, and our own Canadian virus data portal. There. Okay, so this is a little bit about how genomic surveillance is actually performed. We have the genomic sequence data, and then we have signals that are associated with those genome sequences. Like I mentioned before, they may be associated with therapeutic failure, diagnostic failure, increased severity, increased transmissibility, vaccine escape, or natural immune escape. And those are the things that we want to watch and associate with our genome sequences. So we perform an assessment, and this goes on all the time. It's a regular activity. We're always doing this. And then, depending on our assessment, we will have a public health response. And so that may result in a shift in our public health policies, like I mentioned, things like closing the borders, or changing things like quarantine status, or the choice of the therapeutics that we use to help treat the disease. And then also communications back out to our provincial and territorial partners. That's what PT means, and also the public health agency. So we have a group called the Federal SARS-CoV-2 Variant Surveillance Group that monitors the signals and performs that signal strength assessment. If we find that the signal is pretty strong, we will hand that over to our risk assessment group, and they will perform a risk assessment on that specific variant. Now, I didn't have authority to publish any of the risk assessments that we generate, because these are, well, they're internal documents, but there are other public health authorities like the UK Health Security Agency that do publish their risk assessments for certain variants. And so this is an example of what a risk assessment looks like. Essentially what you're looking for are transmissibility, severity, and some of these other indicators like that I've talked about, if they're relevant. So this is an example from the UK HSA for a risk assessment for the BA2 variant, which is a sublineage of Omicron. And ours look very, very much the same. Okay, so just a little bit on international performance. So our national SARS-CoV-2 genomic surveillance program, actually one of the strongest genomic surveillance programs for SARS-CoV-2 in the world. We're top 5 globally for the numbers of sequences that we share up to the National Archive, to the GSAID archive. This is despite Canada having only 0.5% of the global population. And our median time to deposit over the last 13 days is 14 days, so 2 weeks, which I kind of mentioned before. And yeah, we've submitted close to 400,000 sequences out of about 450,000 sequences that we've generated so far. And we perform very well. So and I'm citing this recent article that was looking at essentially doing a performance analysis of national SARS-CoV genomic surveillance and data sharing, and Canada does very well. So we got top marks for high level of routine genomic surveillance. It says we are doing it routinely. High availability, which is our ability to be able to detect a new variant at a 1% or less proportion within the population, and we can do much better than that. We did really well for the proportion of sequences or portions of cases that are sequenced. So we're about between 10 and 15%. So that if you have gotten a test, SARS-CoV-2, if you have a SARS-CoV-2 test and you were positive, there's about a 10 to 15% chance that that specimen has been sequenced. So and so we're very strong there, one of the highest in the world. And then also for the cumulative number of genome sequences that we contribute. Like I said, we're 5th globally. So so it's a pretty impressive program. But that's enough about the program, and I want to move now a little bit more into some of the visualization and data analytics. And so to do that, I'm going to start off with the methodology that we use to track transmission with genomics. And so that is summarized here with this little infogram. So I think it's pretty familiar. A virus infects a person, it will replicate within that individual. And then the virus is passed on to new individuals. And then that whole process is repeated. But sometimes the virus will mutate. And if that if you sequence that mutation, then you can identify possibly like the source or the pattern of transmission for that mutation. It links the transmission chains together. So and so that is essentially how it's done. Now let's take a look here and see, take a look at it in a different way. So this is a toy example, but here's an example of a virus that is initially detected and sequenced. And in this case, I'm looking at the Wuhan 1 reference sequence, which is the first genome sequence that was published in the public archives up on NCBI. So one of the first viruses, and that's like a reference sequence. So that one was sequenced, it was sequenced in China, but over time, it will transmit, and we know this because we've seen this happen, it transmits to other regions and may acquire a mutation. And that mutation may be specific to that region. So it is linking the region at a certain period of time, and it's linking it to its ancestor. So the mutations link the pattern of ancestry and the epidemiological information, the place and time. Gives us that combination is what we call genomic epidemiology. That's what is allowing us to be able to do this type of transmission tracking, and we can build a tree. And so you see over on the right here, we've got a small little tree. It's just a sapling right now, but wash, rinse, and repeat, and you start to build bigger trees with more location and temporal information and mutation information that you link together. You combine all that information together, and that allows you to track the transmission of the virus. In the end, you end up with these big bushy trees, these large phylogenies that I'm showing you at the right, and these messy transmission chains, which can be hard to disentangle, but the information is there. And the idea is you don't look at a global phylogeny, except for in some circumstances that I will show, but usually you're just looking at little parts of the tree and using that to infer how it may be spreading. There. Just a bit of a digression though before I move back to phylogenies. It's also, in addition to tracking the transmission, it's also important, and this is what genome sequencing gives us, is the ability to track the mutations and identify which mutations may be important for things like vaccine escape or disease severity. And so I'm showing you these 3 variants of concern, alpha, beta, and gamma. These arose in the fall of 2020, and they really started spreading around in early 2021. So, and these are molecular models with mutations shown as the red molecular regions here. These all arose in different parts of the tree. So they're all diversifying independently of each other. But when they do emerge and you analyze these genome sequences, you can identify that they have common mutations, which means that they have independently acquired the same mutation, and that mutation is fixed. It's preserved, and so it's referred to as convergent evolution, and it gives us an idea that there may be a selective advantage for that mutation. So for example, alpha, beta, and gamma all have the N501Y mutation. So this is in the spike protein. Actually, it's in the ribosomal binding, it's in the receptor binding domain up here at the top that binds the ACE2 receptor on the host. So that already gives us an indication that it might be important. But the fact that it independently arose in these 3 different variants of concern, which all have higher transmissibility, gives an indication that this may be a mutation that is important for transmissibility. The K417N also resides in the receptor binding domain. It's not present in alpha, but it's present in beta and gamma. So these ones independently acquired this one, and these are associated with immunescape. There's also this E484K mutation that's present in beta and gamma, but is not present in alpha. And this is also strongly associated with immunescape. So this is this additional important information that we use to understand how the evolution of the virus is impacting the epidemiology and the biology of the virus. Okay, so here is an example where the global phylogeny is important and can give us some interesting insights here. And so I'm still showing you specifically, well, it's a global phylogeny, but it's showing you the density of mutations on the left here in the spike protein. So you'll see in the bottom left, over in this section here, where we have like gamma and delta, beta and alpha, they all group together, but Omicron here is way up on its own here. So we're tracking the number of mutations on the y-axis here. And then this is the divergence, number of mutations away from the root. And so you can see that there's this big, they form their own, what's called a clade. It formed its own clade, and it's very much separate from these other variants of concern. And there's a circular plot that shows the same thing here. So within the Omicron lineage, there are the Omicron variant of concern, these 2 sublineages. One's called BA1 here, and that's this golden color, and then BA2, and that's this light orange color here. So there's these 2 separate clades, and we see that they're very much separate from the rest of the tree. They have what's called a long branch length to its most recent common ancestor. This is the earliest ancestor that we can identify that it was linked to. And so this tells us information about what may be going on. What we can say is that it's showing a pattern of unsampled diversity for a long period of time, actually for about 1.5 years, where none of the ancestors of the Omicron lineage were observed. So they just kind of arrived and jumped onto the scene really suddenly. Actually, in South Africa is where they first emerged, and then quickly spread. But this pattern gives rise to certain hypotheses that we can generate. Essentially, one is that we just didn't sequence it and we're unlucky, but it's been spreading the whole time. Cryptic spread that's what that's called, but that doesn't make a lot of sense because there's enough sequencing going on, especially in South Africa, that it would have been detected before then. So we have unsampled diversity for Omicron, and that's what this tree is telling us. So what can explain unsampled diversity? One hypothesis is that it spills over into the animal population, and it evolves in the animal population, and it spills back. And there are examples of that happening. Mink and white-tailed deer both have evidence of a spillback into the human population, although it has never established in the human population and propagated. The other one hypothesis is that that a long-term carrier, possibly immunocompromised individual, they harbored the virus for a long time. They can't kill it completely, so it manages to survive inside that individual. It doesn't kill the individual, but the individual cannot kill the virus. So it allows that virus to evolve within that individual. Actually, it accelerates the evolution in that individual, so it can acquire more mutations relative to the general population, and it can adapt itself to escape the immune system of that individual. So it's learning how to transmit better and how to avoid immune system attack. That is the current hypothesis. This means it is just a hypothesis. You can't prove it, but that is the current hypothesis for how Omicron may have diverged. So here in Omicron, there's a long branch length, and that is, you can see that branch length here, back to the root over here, or back to almost to the root. So it's an ancestor of a lineage called B.1.1, and that was last seen in about February of 2020. And then the hypothesis, that person contracted it and then harbored that virus, and it diversified over time. Essentially, it is diversifying within that individual the same way that SARS-CoV-2 diversifies in the population. It's just unsampled diversity because we're not sequencing that individual. And the BA1 and BA2 lineages both popped up onto the scene at the same time. So it's likely that the BA1 and BA2 variants diverged within that individual. So they have diversity that they are transmitting. When there is a transmission event, they transmit both of these variants, BA1 and BA2, in different proportions. And it's likely that the BA1 was the dominant variant and the BA2 was the minor variant, but they get transmitted, say in a super spreader event, people will contract and propagate one of them, and then they will contribute that back on. And that is likely what we see. This hypothesis explains what we see, where you don't see any BA1 or any BA2, and then suddenly see them both occurring together. Okay, so that's one important way where visualization helps us to provide insight onto the possible origins of the emergence of variants of concern. But it's difficult to use trees all the time to perform genomic surveillance. So what we do is we will partition the tree up into subsets, and we will provide them with labels called lineages. So I've already alluded to them, like the BA1 lineage and the BA2 lineage here. Once you assign the parts of so the tree is diversifying, parts of it are growing, and parts of it may be shrinking, but as it proliferates and diversifies, you can tag them with these labels like BA1 or BA2, BA2.3, whatever, and this will allow you to monitor that lineage over time, like I have shown here in the bottom chart. This is a history of the major lineages in Canada over time, but also pointing out BA1 and BA2 over here. So this is a kind of a toy phylogeny here that we're looking at, but the BA2 branch here, you can see, has just these 3 lineages here. It's small at the start, and there wasn't very much BA2 at the start. It was the minor lineage at the start. BA1 had lots, and so there's lots of subdivisions in that tree, but then over time, so this is the early phylogeny, here's a middle phylogeny, the BA2 ended up overtaking BA1, and so the BA2 starts, it's proliferating, diversifying. New labels are being added to that part of that branch of the tree, but because there's not any more proliferation of the BA1 branch, you don't get any more. There's nothing new to assign. Over time, you see how this is just stagnant over time, but the BA2 continues to proliferate, and new lineages are assigned to the growing branches of the tree. So that's essentially saying that there's nothing left here to assign anymore. This is the one that's taken over, and now we're assigning new lineages to this part of the tree. So this by assigning lineages allows us to get away from having to work with trees all the time and interpret trees, which is difficult, and use simple charts, which are much easier to follow and to interpret. Okay, so how do we do lineage classification? Well, the first step is to build a tree. And the way that we build a tree is by taking the genome sequences and we extract out the variation, all the mutations that just differentiate the given sequence from the reference sequence, that Wuhan 1 reference sequence that I've referred to earlier in the presentation. So you use those mutations, the diversity, to infer a phylogenetic tree. So here's a phylogeny that's built from these genome sequences. Then you start to partition up that tree into subtypes here. So you're designating lineages based upon either there's something important epidemiologically, like it is identifying a lineage that is associated with a certain event or with a certain region, or a certain transmission event, or it may be some type of genomic or biological significance, like it just forms a distinct clade within the overall phylogeny. So you identify that lineage. We use the system called pangolin, which is a dotted decimal notation. So it is recording the placement of that cluster within the larger phylogeny. So for example, here's B.1.5. That is a descendant of the B.1 clade, which itself is a descendant of the B clade, right? That's what a dotted decimal notation does. Is as you add more dots, you're essentially just repartitioning clusters into smaller subclusters. Okay, but there's over 11 million genomes in GISAID. There's actually about 11.5 million genomes in GISAID. There's really a ton of them. And over, I think this slide's a little bit out of date. I think there's probably closer to about getting close to 2,000 lineages that have been assigned. It's a big job, a big process, and not easy to do. So manually, so we use these automated pipelines to help us out. This is the Pangolin suite of pipelines is the internationally adopted pipeline for assigning genome sequences to their lineage. And there are two main ways to do that. One is with machine learning using a system called Pangolin, and we'll talk about that. Then there's another one called Scorpio, which uses constellations of mutations, and it is more designed to identify variants of concern rather than every existing possible lineage, because variants of concern are really important, so you want to get them right. So it's kind of a backup for the Pangolin machine learner, and you use that combination to assign the lineage. And this is ongoing. The Pangolin lineage assignment scheme is a dynamic assignment scheme, so it will it changes over time. As the tree matures, it may require some adjustments to the lineages, how they're assigned, and also there are new lineages that are introduced over time as the tree diversifies. I showed you that in the previous slide. Okay, all right. So, and that's what this slide here is saying, is that because there's a massive number of sequences, not computationally feasible to build a phylogenetic tree every time, take your new sequence and place it in that phylogeny, and then assign a lineage to it. You want to have a faster method, an automated method that is more efficient and also standardized, so that when other people, so if the two sequences are exactly the same but sequenced in different labs, say in different countries, they're going to generate the same lineage at the end. Yeah. Okay. So the method is to perform some training on a subset of the total number of sequences. These are the curated sequences and their labels, essentially their lineage. And then you use that trained model to predict the lineage of a newly generated sequence. Okay, and the type of machine learning model that Pangolin uses is a decision tree. And I don't think I need to explain in too much detail about decision trees to this group, but decision trees are, it's a type of machine learning where you are looking at the features and then using those features to make a decision on the classification in a kind of an iterative and easily visualizable way. So you have the root, which is where everything starts. You have these internal nodes, and these are they will split the tree into subtrees, and then you will and then you'll continue working down your splitters until you get to a leaf node. And then these are the decision makers. These are the ones that will actually decide the class. So this is what a Pango machine learning model looks like. It's maybe a little bit hard to see here, but this is it's a decision tree that from a while ago, but visualized. Here's a kind of a blow up of it to kind of see what it looks like. So here it's saying, look, at position 18423, there's a gap here. If that is true, then you'll go to this node. If it's false, then you go to this node, and then wash, rinse, repeat until you work your way through the mutations that you have and this decision tree to assign a subtype at the end. Okay, one of the problems with decision trees is that they have this risk of overfitting. So they will do a really good job of classifying what they've seen in the past. They don't do such a great job of identifying new sequences. So that's this overfitting problem. The way to help to ameliorate that problem is to use random forest. So the Pangolin machine learning model used to be a single decision tree. Recently, it switched to random forest. So a random forest is just a collection of decision trees where you randomly select a subset of the training data, say 80%, right? And it's a random 80%. You just do that over and over and over again until you build a bunch of trees, these subtrees, and then you will use the classifications from each of the subtrees to, well, each tree will make a vote on, look at the features and then make a vote on how to classify it, and the majority vote wins. And so this helps to ameliorate the problems with overfitting. Okay, so Scorpio is, I mentioned before, it's used more on a small subset of different variants, the variance is concerned, and it uses constellations of mutations. So, so here's some code that shows how Scorpio does classification for the alpha variant of concern, which is B.1.1.7. This is extinct now, but this is an example here. So there are 23 mutations that it is looking for. And if it says, if I see a certain number of those mutations, the rules here are that it has to see 15, and you can't have more than 3 that are reference of the mutations in this set that are what we call wild type mutations or reference mutations. There, yeah. And so there, and so about 65% of the mutations that the signature mutations in this constellation have to be detected in Scorpio to be able to call the alpha variant. Okay, so it's a kind of a backup system for making sure that you didn't make a mistake when trying to classify a variant of concern. There. So I mentioned the Pangolin system is dynamic, and so the classification changes through time. Well, depending on whether the tree is, the shape of the tree as it evolves over time may require some changes in the classification scheme, or you may need to add new lineages over time. It showed that. And so there are changes that do happen with some of the existing lineages. That's what's meant by dynamic system. So every release, there's going to be some changes. And so we need to track that because that has implications for genomic surveillance and so that we can understand what we're seeing, especially when we upgrade our pangolin machine learner. So there's an average of about 4% change between consecutive models over time. Here you can visualize it, and we do visualize it. This is work that's done by Dr. Julie Chen, who's my lead. I'm my head of data sciences in my lab. So she developed this Sankey plot here that allows you to see between iterations how are the existing lineages being repartitioned up into sublineages and how are they changing. So this is an example where we had one release from late 2021, and then another release that came out a couple of days later. And how did they, you can actually visualize to see how did these lineages get reassigned between these releases. Now, it's also important to take into account the hierarchical nature of these lineages. So we want to know not just did they change, but how did they change? Because the trees are hierarchical and the classification system is hierarchical, there are a number of different types of changes that can happen. One change could be to revert a child back to the parent lineage, or the other way around, where a parent is partitioned up into a child. There's also sibling lineages. So BA1 and BA2 are sibling lineages. And this is important for us to want to take note. So every time we do an update, we will perform this analysis to see how, what are the implications for how things are changing within our existing proportions of circulating lineages in Canada. So here's an example where AY.45 was changed to AY.74. So that's a sibling lineage change because they're at the same point within that dotted decimal notation, 45 and 74, rather than like a 45 to 45.1 or 45.1 to 45, which would be a child to parent or parent to child type of relationship. These are sibling relationships. And so, and what we did notice in this specific example is that there was a large number that were being designated AY45, and that is a South African lineage, that these were redesignated to AY74 because a lot of the Canadian lineages were being misassigned to this South African lineage just because they're similar and the classification scheme didn't quite get it right. So, and we communicated that with them, so they made this change so that it is better representing the lineages and their association with the region of origin, which is Canada in this example. Okay, so nowcasting, that's another. So nowcasting is taking the data that you have and trying to forecast it for a short period of time into the future. And this is done, it's taken from economics, but it is applicable to predicting how the proportions of circulating lineages may be changing in the future. But because there is a lag time in the data that's collected, so remember, we have about a 2 to 3 week lag time, closer to 2 weeks lag time between the time a sample is collected and the time that it is submitted to the national database or to the international archive. So about a 2 week period of time. And what nowcasting does is allow us to fill in the blanks here. So this is an example on the left of the US CDC, which they publish their nowcasted data actually as default. So here it is with the nowcasting off, right? And so the most recent date here that they report is, well, that would be May 28th, right? But when they nowcast it, they can predict it much closer to the current date. Here they're predicting it up to the 11th. Well, this is the weekend, yeah, the week ending on the 11th of June. So this data doesn't exist. These 2 weeks here don't exist. This is being nowcasted using this forecasting algorithm. And so it's based on, it's a it's a multinomial logistic regression or multinomial regression type of calculation. We are just in the final stages of implementing our own nowcasting. Not sure if we're going to be publishing our nowcasting results, but we have been working on developing some code to allow us to do predictions. And of course, we have to evaluate the quality of the predictions. And we find that it does quite well when you have lineages that exist in a very high proportion. So these are the ones that are up at the top. When they have very high proportion, it does a good job of predicting. So these dots here are the actual proportions, and this dotted line here showing us this is the latest date that we allow the predictor to see, and then everything after that is the data that we have, and we see how well does it predict the real data for about 2 weeks beyond the cutoff point. And so for when the lineages are circulating in high proportion, so the curve here in red is showing us the prediction, and then the dots are the actual, and you see it does pretty well. This one here, it makes a mistake because it drops off right at the peak, right? So it does have trouble predicting right at the peak, right? But other than that, if the peak happens before it's being predicted, it does quite a good job. Does an okay job with ones with lower proportion, but not perfect. So you can see there's high confidence intervals here, these thick bands that make it less useful for ones with low proportion. So we'll be implementing this, but we're implementing it with just for the lineages that exist in high proportion. So here's an example of where we are predicting it in, well, of course we use the prediction and we can check against the actual data. And you can see that it does a pretty good job here. So this is the actual data, and then here's the predicted data here. So you can see, like for this, for BA 2.12.1, 30.6 versus 30.5 is pretty good. 37.4 after the second week versus 31. Okay, it's not doing so well. But for other ones like BA2, 45 versus 43 and 42 versus 44, it's tracking pretty well. So it does it's not perfect, but it does a good job, and it allows us basically to fill in the blanks between that lag period for the data that we have and what we expect is actually circulating today. There. What time is it? You know what? We're just about at the end, and so I am going to skip this, even though I'd love to be able to present this work to you, and just finish up here. So there are other data analytics. Actually, the data analytics was one of the biggest working groups within the this large program, especially in the early days, just trying to get stuff up and running. So and that over 60 bioinformatics, like microbial bioinformatics scientists, have contributed to developing the data analytics for this program over the last 2 years. And so some of the major products are the pipelines. nCoV tools which does all of the assembly and identification of the mutations for Oxford Nanopore data. Signal is its equivalent for Illumina data. Covizu, that's over here. That's a program that is used to simplify the visualization of the diversity that is present in the global phylogeny. So trees are hard to look at. Covizu makes it easier to look at. COVID MVP allows us to assess what the functional significance of a mutation is. So remember, I was talking about E484K and N501Y. Those are, the significance, the functional significance, like what is its impact on severity or transmissibility or biology. This COVID MVP program scours the internet and records everything that's known about mutations, so that if you get a new, soup du jour variant and you want to see like what are the mutations and the significance of the mutations. That's what COVID MVP does. We have our VirusSeq data portal. So this is the kind of like our Canadian repository of SARS-CoV-2 genomic sequence data. Surveiller is this app here. It's a dashboarding app that was built in-house that allows us to track the proportions of circulating lineages in Canada. IRDA is our web platform for storage and management of the SARS-CoV-2 data. So all of those, remember I said about 450,000 sequences that have been generated and exist in the national database. They're actually being, the IRDA is our national database. That is our platform for storing and managing all of that. Genomics Data Index, I didn't get a chance to talk about. Changelings is the system that we use for monitoring the changes in the way that variants are being classified that I was showing you before with Sankey plots and these histograms. Okay, okay, so there are hundreds of people who have contributed to our national SARS-CoV-2 genomic surveillance program. Obviously, I don't have room on the slide to acknowledge every one of them, but I would like to, well, I'd like to acknowledge my lab. I don't have a group picture for the lab. I apologize for that. Here's an org chart on the right. So these are all the members of my lab, and Natalie Knox is my head of data. Well, she's the head of data analytics for the project, but she's also my head of computational biology. And it's largely the people in her group that have contributed to the data analytics that I've been showing you here. I won't go through them individually just for time, but I'll end it there, and thank you for your time.